Using decellularized grapes to grow yeast. In my project, I used the process of decellularization in order to create an organic environment for yeast. By decellularizing grapes and cultivating yeast inside of it, I would be able to recreate the fermentation process in the fruit, but without any of the cell contents inside of it. The reason why it's important is because the idea could be expanded to something like adding a SCOBY instead of a yeast to be packaged up and used for fermentation enthusiasts. How I did the project was create a batch of 5 decellularized grapes for over the course of 2 weeks, take microscope samples to compare decellularized grapes and non-decellularized grapes to get a rough estimate of the amount of nuclei that have been lysed, and then condition the grapes to be vessels of fermentation. Then lastly, inject them with yeast and set them in a fermentation chamber with a CO2 probe in it. By decellularizing fruit and cultivating yeast inside of it, we will be able to recreate the fermentation process in fruit, but without any of the cell contents inside of it. What already exists that is similar to my project is transplanting animal cells such as human cells into organic material such as leaves or apples. Although I used the same process to achieve my end goal, decellularization, my outcome was completely different. What sets my research apart from the currently practiced methods of decellularization is that my process isn't meant for medical purposes, but solely for experimental use and proof of concept. For the experiment, the goal was to decellularize a grape, then determine whether the result is an adequate environment to cultivate yeast. By doing this, it creates an original use for the decellularization process, while figuring out a way to cheaply yet reliably decellularize the grapes. In order to do this, I would have to decellularize the fruit, create an environment for the yeast to grow in that the grapes would be monitored in, and condition the grapes for fermentation. Add yeast, then monitor fermentation with a CO2 probe. For materials, I had an array of organic, chemical, and mechanical elements. For produce, I had red candy snap grapes from my local grocery store. I then just cleared off the skin by cutting it off with a sharp knife so that the chemicals would more easily permeate through the grapes. For making the active yeast solution, I used 1 Red Star dry yeast package, 150 milliliters of tap water, and 8 grams of sucrose. For chemical materials, I used a 5% sodium lauryl sulfate or sodium dodecyl sulfate solution to decellularize the grapes. It is important to note that sodium lauryl sulfate and sodium dodecyl sulfate are the same thing. I also used a 70% solution of ethanol for sterilization after the decellularization process to ensure that the yeast would have an uncontested environment to thrive in. For tools, I used a microscope to take pictures to compare the different samples to each other. I also used 4 vernier CO2 probes for monitoring the fermentation progress. For a dye to try to get an estimate of cells while comparing decellularization samples, I used methylene blue. Lastly, I used a normal stir plate to keep the sodium dodecyl sulfate from setting on the bottom of the glass I was soaking the grapes in. The process for decellularizing the grapes was simple, but time-consuming. First, I made a solution that would be used for decellularization with a chemical named sodium dodecyl sulfate, or also known as sodium lauryl sulfate. For short, it is called SDS. I needed to make a 5% SDS solution in distilled water for decellularization. Once I had that, I got my grapes from my local grocery store and skinned 5 of them. After that, I allowed all of the grapes to soak in 250 milliliters of the solution for a week. After the first week, I took a grape out once every 2 days until they were all gone. As soon as I took them out, I put them in regular distilled water in order for the SDS to flush out. I also took grape 2, 3, and 4 to soak in a 70% ethanol solution for a day, so I could sterilize them and see if that impacted the competition between the yeast and other foreign fungi and bacteria. Once all the grapes were cleared of chemicals, I took samples of them and checked for the decellularization progress on a microscopic scale. After the grapes had the SDS and ethanol flushed out, I took my grapes and soaked them in a 10% sucrose solution for about 3 days. Then I made a yeast solution with 150 milliliters of tap water, 8 grams of sucrose, and 5.5 grams of unactivated yeast. For making a suitable fermentation vessel, I grabbed 4 clean plastic bottles that the CO2 probes could fit in through the top. Then I soaked cotton pads with water and set it at the bottom of the bottle. After this, I cut a small section from cellophane and unfurled it on top of the cotton pad. I repeated this 4 times for each chamber. Once I had my fermentation chambers complete, and along with the yeast and grapes, I injected 0.5 milliliters of the solution into 4 grape samples each. After that, I set the samples into the 4 prepared fermentation chambers and capped them with 4 different CO2 probes. Throughout the course of 7 days, I collected data into Logger Pro and created a graph of the change in CO2 concentration. For my results, I achieved everything I had set out to do, but not completely. For decellularization, it was too difficult to try and estimate the amount of nuclei in each sample because the dye I used, methylene blue, didn't exactly penetrate the cell walls in a way that would give us definitive numbers. On the bottom left, there are 2 pictures. You can see dots that are most likely to be nuclei, but it is entirely possible that they are simply artifacts. You can, however, see a big difference in cell structure. The leftmost picture is a sample of a non-decellularized grape, and to the left of that, a decellularized grape. There is a clear, dense, rigid structure of the cells in the leftmost picture, where there are less destructured cells on the left photo. This demonstrates that the decellularization process clearly worked, but even still, there are intact nuclei in both photos. For the 2 pictures on the right, the picture on the left is of unactivated Saccharomyces cerevisiae, straight out of the commercial packaging. On the right is a sample from grape number 4. In the sample, you can clearly see that there is developed activated yeast that is still alive, and also that the yeast has spread throughout the entire grape, whereas yeast on the left is densely packed and is only a tiny bit green if you zoom in enough. In this graph, you see grape 1, the blue line, and grape 2, the red line. You can see large fermentation from hour 1 to 17, and that the grapes clearly did somewhat ferment. But then, after hour 17, it drops dramatically and stays consistently at a baseline level of CO2 production. It is important to note that the grapes had no major visible change after hour 17, other than the oxidation of the grapes. In this graph, you see grape 3, the red line, and grape 4, the blue line. The same thing goes for this graph, but the main difference is that the drop was at hour 18 rather than hour 17. In the end, the project was successful, but can be improved with gained experience. One thing I could have done is allow the grapes to soak in SDS for longer. I also messed up the ideal ratios for yeast activation, but if I would have done more research on it, I would have gotten it sooner and gotten a better product. More food for yeast probably also would have yielded a better result too. It did, however, turn out to be an effective vessel for yeast, though. There was no competing bacterial or fungi growth. It did ferment, if only just a small amount, and it was easy for the yeast to fully spread. Some way I would build off of this project would be to replace the yeast injected into the grapes with SCOBY, and make a shelf-stable product that would be able to be packaged and stored for prolonged periods of time. Another thing I would do is to determine what caused the yeast to stop growing, and also determine alcohol levels in the samples. How I would do the project differently is by having more time to experiment with the processes. This way I would be able to test equipment for consistency. I would also have acquired a dye that accurately shows the amount of nuclei, as the methylene blue didn't give much definitive data.